Good morning students. Today we are going to discuss about fermentation technology. This fermentation. Okay, so here the fermentation means it's a Latin word which means fermi means to boil. Fermi is a Latin verb which means to boil. And this is arise or this name is given because whenever the fermentation takes place, the boiling appearance, that is the bubble arises from the bottom. Say for example, whenever we boil the water, when the water reaches its boiling point 100 degree, it starts bubbling from the bottom. Have you seen it? So in such a way, whenever the fermentation takes place, the release of carbon dioxide occurs. So whenever the carbon dioxide releases occurs, it starts boiling, it just starts rising from the bottom as like a bubbles to reach the top. This is due to the metabolism of your carbon or organic compound which is present in the substrate. Okay, so that the same condition occurs, that's why it is called as fermentation. In simple, in biochemical method or in biochemical meaning, it is given as the energy production process. That is energy production process by means of catabolism of organic compounds. That is by means of biochemical, it is simply said as generation of energy by means of catabolism of any organic compounds. Okay, so this is all about the basics of fermentation, and we doesn't know the history. Say for example, right from our ancient days or ancient times, we have started drinking all the alcohol, that is wine. So that is produced by means of fermentation. So right from the Rig and Yajurveda itself, this fermentation technology is used. But exactly by 1940, this 1940 is important. That is right from 1940 to 1950, the industrial revolution takes place. That is by 1940, what is introduced? That is the penicillin. Penicillin is an antibiotic which has been produced through fermentation process, which has paved way for all industrial revolution process. Because during the World War II, whenever the people have started dying, this penicillin antibiotic is given so that this started saving the huge number of people's life. So from that, the industrial revolution takes place. Till then, the industrial production process is limited. But during this, they started investing money on the penicillin production. Whenever some owners are investing, they always think to get it back double or triple. So then, when the insulin production started by means of fermentation process, obviously the industrial revolution takes place. So now we will see the ranges of fermentation. That is, this ranges, which means the commercial production of fermentation process. There is 5 major classes or 5 major commercial process or ranges of fermentation. That is those which produce single cell protein, and the second one is microbial production, microbial enzyme production, and the third one is metabolites. And fourth one is recombinant products. And fifth one is transformation. That is, the fermentation process can be produced in 5 major ranges. That is, the first one is single cell protein. That is, the microorganism is produced in large quantities only for commercial production of single biomass. So the single cell protein is a single biomass is produced. So this biomass may be a bacchus yeast which is used for any alcohol production or else for baking industries, or at the same time as a supplement protein for human consumption. So the fermentation is done only to produce your single cell protein. This is one range. And the second one is microbial enzyme production. When we talk about a microbial enzyme production, these enzymes can be produced by either plant or animal or by microorganism. Why we are specifically talking about microbial enzyme production means, since when we get an enzyme from plant and animal, we get it at very less quantity, but whenever you are going for a microbial production, you can cultivate a huge number of enzymes. That is, you can harvest a huge number of enzymes that the major source from microorganisms. So we can produce a large quantities of enzymes. This process will be economic and cheap. And third one is metabolites. Here the metabolites are majorly classified as primary metabolites and secondary metabolites. That is, during the growth phase of any microorganism, that it is called as log phase, and from exponential phase to stationary phase, this secondary metabolites are produced. So the primary metabolites is mostly used for growth, and the second one is for maintenance. Okay, say for example, all your organic acids, alcohols, vitamins, antibiotics, or the metabolites. And the third one is recombinant product. When we talk about a recombinant product, that is the foreign enzymes or the higher genes of higher organisms are introduced into the microorganism that act as a host, and so that the microbial recombinant products are produced. And the transformation is microbial cells, which is used to convert a compound of structurally related one, that you can go for via biotransformation process. That is, this can simply set by means of triangle value. That is, when the biomass value is high, okay, this is what single cell protein, and this is enzyme, and this is metabolites, this is recombinant product, and this is transformation product. Here the mass production is high and the value is less. As the number of as the quantity production is less, the value of the product is increased. Okay, so say for example, the single cell protein, as said earlier, it is only used for baking industries or as a supplement. Okay, when it is used, it is cultivated in large quantities, but the value is cheap. At the same time, the transformation product or recombinant product, it is produced in less quantity, but for the purification process, at most 100% importance has to be given, because it has been consumed or to treat any disease. So for that, you are spending a lot of money. So in turn, the cost or intern cost what you are receiving is high. So the quantity of production is less, but the yield is, in terms of the amount, it is high. Okay, so this is what the ranges of commercial production of fermentation process. So now, what are the major requirements of any fermentation process? So ultimately, the vessel, which is called as fermenter, which is required for any fermentation process. Okay, now as like your both the eyes, right eye and the left eye, for any fermentation process you need to have the microorganisms and at the same time media. Okay, so here the fermentation process can be split again in two ways. One is like upstream processing and the next one is downstream processing. Okay, so here the upstream processing is till the fermentation takes place, it is called as upstream processing, and after the fermentation to derive a final product, it is called as downstream processing, that is below the fermentation process. Now as said earlier, I am talking about left eye of the fermentation process, that is what your microorganism. Now just for example and easy understanding, I am stating that alcohol production, okay, or ethanol production. So there are n number of microorganisms is capable of producing ethanol. So the best of it is Xanthomonas mobilis or Saccharomyces cerevisiae. So whenever you know that whenever we are writing microorganism name, it should be written in italics or you should need to underline the microorganism. now Zymomonas mobilis, which is capable of producing an alcohol ethanol. Okay. Now say for example, there are again, this is what the genus, this is species. Okay, so as per the classification, genus, species, there is n number of species available in the Zymomonas. So out of which 10 species, which species is capable of producing higher yield of ethanol, I need to check. Okay, so then I have to fix one microorganism which is capable of producing an high yield of ethanol. Okay, so in this one species, I have to alter the gene sequence if needed. Okay, which can withstand drought conditions, or even when the nutrients are less, it could survive. So by means of recombinant DNA technology, I can remove certain genes and I can fix the required. So now I have fixed all this required gene. Now this I have to process. Okay, that I am just checking out this microorganism and processing by means of or mutation and strain selection. A suitable strain is now selected and I am preserved it. The reason why I am preserving and making it as stock culture means, say for example, now I have used the best culture of Zymomonas mobilis and produced ethanol and I have got maximum yield. But after certain days, if I could not able to get the Zymomonas mobilis where I have isolated, means I will always in a trouble. So when I put this as a stock or mother culture, I can preserve it for longer time. When I preserve it for longer time, I always take a pinch, I can do subculture and I can reuse it. So for this process, I have to go for stock culture. This is what a microorganism. So I have to isolate the microorganism and I need to identify which microorganism is best and suitable for alcohol production, in which. So I have to check which microorganism is capable of producing maximum yield. From that I have to develop further strain development, and I am doing all genetic engineering work to withstand and reduce the byproduct formation. Finally, the stock or mother culture is obtained. Okay, so this is all about microorganisms. Now I need to talk about media. Okay, so when I talk about the right type media for the fermentation process, in our laboratory in general, what we do, we use all synthetic chemicals. The synthetic chemical means it is costly. So here, for your example, the 500 grams of nutrient agar is 750 rupees. So for example, you use 2.3 gram per 100 ml. Okay, so 2.3, and if it is of 1 liter, it is 23 grams. So for 1 liter, you are using 23 grams, which means approximately 2 rupees to 3 rupees. Okay, if this is a condition, in industrial fermentation, you use lakhs and lakhs of liters of water, so that you have to use kilograms of media. Then it is not easy and viable for any industrial process. So during the industrial fermentation process, we go for raw substrates. That is like molasses, paddy stock, wheat stock. But for maintaining the stock culture and for isolation, you use the synthetic media, right? So for initial process or for a less quantity, you use the synthetic media. But whereas when you go for commercial production, you go for raw medium. Okay, so then you have to check whether the raw medium consists of carbon source, nitrogen source, phosphorus, and desired pH, viscosity of the medium is good or not. So all these things has to be done and it has to be taken into consideration for fermentation. And important thing is, this medium you cannot use directly because it has lot of contaminants. So it has to undergo sterilization process. So then again you need to check whether the medium is capable of undergoing sterilization, because whenever the sterilization is taking place, Brownian reaction will take place. Brownian reactions. So this is also technically called as Maillard reaction. So when the Maillard's reaction is taking place, that is the substance called amino group of the amino acids and nitrogen group of the another amino acids, when it combines on heating, it turns the compound as brown. Say for example, best example is like Adarsan sayarappa pagu kachana, it turns as a brown color. So just like that, it is called as Maillard reaction due to overheating. That may occur. If that occurs, the nutrient will get lost. Okay. So all these things is to be considered during the medium formulation. When everything is okay, you come for fermentation. So as we are comfortable right now here and we wear a normal dress, okay, but the same people with the same dress, if we go and sit in Switzerland, it is not possible because our body condition and our cloth will not be suitable. So the same thing for the microorganisms too. So you have to give a proper environment, that is like pH, temperature and agitation for proper mixing of media and microorganism. So all these things has to be given in any fermenter which is fixed, so that the microbes grow and produce a better yield, so that the microorganism can utilize the medium or the nutrient which is present in the medium, it grows and produce the desired compound alcohol. Then after 36 hours of fermentation, when all the nutrients are get exhausted or utilized by the microorganism, then it is ready to harvest. So till now, before harvesting, it is called as upstream processing. So once it is ready to harvest, that is the cell is ready to lyse and ready to get your product, from that it is called as downstream processing. Okay, is it clear?